Hi everyone and welcome to Occulus Labs. In this video, we are going to learn about a very important and a fundamental staining used in microbiological lab to differentiate the bacteria into two broad categories, gram-positive and negative bacteria. So first of all, we will learn about the bacteria, like what is the principle and what are the cofactors that play a major role to decide that whether it will be a gram-positive or negative bacteria. So let me tell you first, like here we have a gram positive bacteria. You all need to know that bacteria are the single bacteria are having only a single cell. Okay, so both gram positive and gram negative will have a single cell, and that staining we are learning about gram staining, it only works because of the structure of cell wall in gram positive and gram negative bacteria. So we do not need to learn about this inside the cell and other organelles which are present inside the bacterial cell. It is not our part. We have to learn about the structure of cell wall so we can decide that how the gram staining works. So first we will learn only about the gram positive and gram negative bacteria cell wall. So what is the cell wall? Every cell has a protective layer that is called a cell wall. Like in our human beings, we see that we are having the skin that is very thin, but also bacteria are having a protective layer that protects their cell, that is called cell wall. So in gram positive and gram negative, both are having a cell wall like that. This is the cell wall of bacteria. Okay, so they both are having cell wall to protect their cell because they are having only one single cell. If it will be destroyed, their whole future will be destroyed. I hope you got it. So now gram positive and gram negative both are having cell wall, but the difference is that gram positive bacteria is having a very thick layer of peptidoglycan. What is that? That is peptidoglycan. Gram positive are having a very thick layer of peptidoglycan, but in gram negative you see that the layer of peptidoglycan is thin compared to gram positive. And additionally in gram negative you will see that they are also having a smooth outer membrane made up of lipopolysaccharides. So in gram negative they are having two structure. They are also having a peptidoglycan layer, as well as they are having a thin outer membrane made up of lipopolysaccharide. But in gram negative you will see that they are just having a thick layer of peptidoglycan, which make approximately 50 to 80% of their whole cell wall structure in gram positive. But if you will see in the gram negative that the peptidoglycan layer only takes up to 5 to 10% of whole cell wall structure. So this is the major difference between gram positive and gram negative bacteria cell wall. And because of this difference, the reagents work differently from both the organism. So now if I take it to in a chemical perception, in the peptidoglycan layer there is one polymer called teichoic acid. Inside the peptidoglycan layer you will see that there is one polymer called teichoic acid. It is present in both gram positive and gram negative bacteria's peptidoglycan layer. Okay, and it is negatively charged. You need to remember that teichoic acid is negatively charged, and because of this charge the dyes are combined with the peptidoglycan layers. So you need to remember that this is the main factor that decide that whether it will be a gram positive or gram negative bacteria. Okay, so now in the gram staining we use the primary stain. So what is the primary stain? It is crystal violet. Okay, so this is the crystal violet. So when you use the crystal violet, like in suppose for 1 minute we use it for gram positive as well as for the gram negative bacteria. So when you use in the gram positive bacteria, the crystal violet is positively charged dye. Okay, so negative and positive will be combined with each other. So in the gram positive, the crystal violet will go deep inside the peptidoglycan layer and will combine with the negatively charged polymer and stain the cell wall of gram positive bacteria to purple. Same thing will also happen in gram negative, but because of our outer membrane, this outer membrane works as a barrier. This, and because of this membrane and a thin peptidoglycan layer, crystal violet combines with the teichoic acid very loosely. But after the crystal violet staining, if you observe the slide into the microscopy, you will see that both gram positive and gram negative bacteria are looking purple because of the crystal violet and negatively charged teichoic acid present in the peptidoglycan layer of the bacteria. Okay. So after the first step of the crystal violet, there will be no difference between gram positive and negative bacteria. And now the second reagent is what? The second reagent we use is iodine. Grams iodine is what? It is grams iodine. Okay. So what is a grams iodine? Grams iodine is not a dye which is used in the staining. Grams iodine works as a fixator. The work of grams iodine is to fix the crystal violet inside the peptidoglycan layer of gram positive and negative bacteria. It's a fixator. So it goes inside the peptidoglycan layer of gram positive and negative bacteria and will combine with crystal violet, okay, and will form a chemical complex. And what it will form? It will form a chemical complex. That chemical complex will be trapped inside the porous structure of peptidoglycan layers. So let me remind you again that gram positive is having a very thick layer of peptidoglycan. There are multiple layers of peptidoglycan. So that chemical complex of crystal violet and grams iodine will be trapped very deeply and will have a tight bond. But in gram negative, you will see that because of the outer membrane which works as a barrier, and also because of the thin layers of peptidoglycan, the bond of crystal violet and grams iodine chemical complex with the peptidoglycan will be very loose because they are only having few layers of peptidoglycan. But in gram positive, you will see that there are multiple layers of peptidoglycan, so that will be a very tight bond between the chemical complex and the peptidoglycan layers. And here comes the twist. The third pass is the most important, and I would say it is the main deciding part in the gram staining procedure. So the third part is decolorization, okay? It's the third part of decolorization. The third part is decolorization. So as a decolorizer, you can use either the alcohol or the acetone, whatever you would like to use, you can use it. But I would prefer that you should use the acetone because it takes less time and it is very effective. So when you use the acetone for the gram positive bacteria, because of thickness of peptidoglycan layers, it won't be able to dissolve the whole thickness of peptidoglycan layers. So the effect of like acetone in the gram positive bacteria will be zero. Acetone will not be able to impact anything in gram positive because of the thick peptidoglycan layers. So after the decolorization, you will see that gram positive bacteria are still looking purple. But as you apply the same acetone on the gram negative bacteria, you will see that the membrane of lipopolysaccharide will be ruptured because of acetone. This, it is ruptured. As soon as it is ruptured, the thin layer of peptidoglycan will release the chemical complex of crystal violet and grams iodine out of the cell wall. It will be released, okay? So after decolorization, you will see that gram negative are looking colorless. Why it is looking colorless? Because of the acetone, all the chemical complex of crystal violet and grams iodine which was present inside the thin layer of peptidoglycan, it is leaked out. So after the decolorization, you will see that it is colorless and the gram positive is still purple. And now comes the final most part, is the counter staining. So as a counter stain, I am using carbol fuchsin. You can use the safranin or carbol fuchsin, whatever you would love to use. But let me tell you again that carbol fuchsin or safranin is the positively charged dye. So here comes the chemistry again. So in gram-positive bacteria, negatively charged polymer teichoic acid are already in the bond with crystal violet, which is again a positively charged dye. So there is no space left for the negatively charged teichoic acid to take part in the again with a positively charged dye. So there is no place for the teichoic acid. There is no free teichoic acid polymer which can bind again with the positively charged dye. But here in the gram negative, after the decolorization, you see that all the negatively charged polymers are free from the positively charged crystal violet. They are free because the crystal violet and gram iodine complexes are leaked out. So they are empty. They are sitting idle and they are waiting for somebody to have a bond with them. Okay, so as you apply the counter stain like carbonyl fusions, the negatively charged polymer will combine with the carbonyl fusion and they will give the color to the cell wall of gram negative bacteria to pink to red. They will give the color to pink to red, but you will see in gram positive they are still looking purple. So this is the main difference between gram positive and gram negative bacteria. In this video we have explained the full principle and how the reagents works and what are the key factors that you need to remember in this. I hope you found this video informative. If you found it, please like and subscribe.